Good morning. Today we are going to talk about the digestive system. The digestive system is one of the many organ systems that we have in our body, which concerns in digestion of the food, chemical and mechanical digestion, and transform the food from more complex structure into more simple form that can be absorbed easily in the small intestine and can be used for energy and for building of different body systems. For the digestive system, it consists and start from the oral cavity, and then we have the alimentary canal. In some areas we have area of dilation. For example, in the human, in the dogs, in the horse, we have the area of the stomach. Similarly, we can see it also in ruminant. Also in the horse, we have another dilation, which we can see in the area of the cecum. So the digestive system consists of the oral cavity, and we have the alimentary canal, which starts from the esophagus till the distal end of the rectum. Also, we have the accessory organs and the extrinsic glands. For example, in the mouth, we have the teeth, we have the tongue, we have the salivary gland, we have the liver, and we have the pancreas. All of these structures help in the digestion process. You have to remember, when we eat the food, it's a complex structure. We have to transform it into more simple that can be easily absorbed and used for energy. And this slide is very important because it describes the digestive system, the component, the histological layers that we can see from the beginning, from the area of the esophagus almost to the area of the colon. The digestive system consists of four layers. We have the mucosa, the submucosa, the muscularis externa, and the serosa. Additional layer, which is the adventitia, and this is the adventitia only found only in the area that any part of the digestive system is not found in a hollow space, not found in, for example, in the thoracic cavity or the abdominal cavity. If it's found in the area of the neck or the distal end of the digestive system, then it's covered by additional layer, which is the adventitia. So we start here. This is the area. So the digestive system from the area of the esophagus to the area of the colon consists, as I said before, from four layers: the mucosa, the submucosa, the muscularis externa, and we have the serosa and the adventitia. So if we start with the first area here, this is the area of the mucosa. It consists of three components. First, we have the surface epithelium, then we have the loose connective tissue layer, which is the lamina propria, then we have the area here. We can see this area here. This is the area of the smooth muscles, and we call it as muscularis mucosa. It's very important that we call it the muscularis mucosa because it's found in the area of the mucosa. So we have the first layer, the mucosa, which is consists of the epithelium and its range. For example, if we start with the esophagus, it's composed of keratinized stratified squamous epithelium, and this keratinized stratified squamous epithelium could be thin or thick keratinized stratified squamous epithelium. If we go to the area of the stomach, then this epithelium will be simple columnar mucus producing cells. If we go to the area of the duodenum and the jejunum, ileum, and cecum and colon, most of these epithelium will be simple columnar with goblet cell. As I said, just below the epithelium we have the layer, which we call it as the lamina propria, and most of the time it's smooth, loose connective tissue. The third component of the layer mucosa will be the muscularis mucosa. You can see here, this area here, this is the muscularis mucosa. The second layer we have here, this is the area of the submucosa, and this submucosa is dense irregular connective tissue. At this area mostly dense irregular connective tissue. Sometimes we have glands, and these glands, as you can see, only we can find these glands in two areas, the area of the esophagus and the area of the duodenum. Then we have the two layers here, which is the area of the smooth muscle, which is the muscularis externa. Very important that we call it muscularis externa so we can differentiate it from the muscularis mucosa. The muscularis externa is two layer, inner circular and the outer longitudinal. If we have this here as the tube of the small intestine or large intestine, we have one layer that the muscle is found as a circular. This we call it as the inner circular, whereas the one that found in the longitudinal axis of the organ, this we call it as the outer layer, which is outer longitudinal layer of the muscularis externa. Finally, we have this area here, which is the area of the serosa, and this serosa is simple squamous epithelium cover the organ, and usually one single cell layer. Sometimes we call it as the mesothelial layer or the mesothelium. As I said, in the area, for example, that we don't have a hollow space, then the organ we have the additional layer, which is the adventitia, and mostly this adventitia is part of the fascia that cover the organ. For example, we have the superficial fascia and we have the deep fascia, and this deep fascia is composed of the dense irregular connective tissue. Again, this is the organ. As you can see here, in this area here, we have the epithelium. Then we have the area of the lamina propria. Then we have this is the area. We have the muscular submucosa. Then we have the area here, which is the area of the submucosa. And then we have the two layers. We have the inner circular and we have the outer longitudinal. Then we have the area of the serosa. So if we start with the digestive system, the first area here, we have the area of the mouth. As you can see here, from outside we have the lips, and the lateral we have the cheeks. Inside we have the teeth and we have the tongue, and we have the salivary glands that mostly they are masses that found outside the oral cavity, but that they have a duct that will open inside the oral cavity. The epidermis that found at the area of the oral cavity, the origin of this epithelium will be from the epidermis, and this is also true for the area of the anus. The epithelium at that area and the epidermis usually the source of the ectoderm. However, the lining of the digestive from the beginning to the end will come from the endoderm, and we are going to talk about that in the course in the embryology. We can see here we have the area of the junction between the lips and the skin. We can see here the skin with the hair, and this area here we call it as the mucocutaneous junction. This is the area of the junction between the mucus and the skin. If we can see here we have the lips, and we can see here this is the area of the oral cavity. This epithelium is the mucosa. Outside we have the skin and we have the hair follicles. We can see here the area just below the epithelium is the area of the connective tissue, and also we can see here this is the skeletal muscle. If you remember from anatomy, at the area of the lips we have the orbicularis oris, whereas for the area of the cheeks we have the buccinator muscle, and mostly these muscles are skeletal muscle because they are very important for mastication. And we can see here this is the area of the mucosa and this is the area of the skin, At the area of junction between the mucus and skin, we call it as the mucocutaneous junction. The second thing that we have on the oral cavity, we have the teeth. The teeth is not part of the skeleton because if you remember, we talk about the skeleton. Usually the skeleton, the bones, the origin of these bones are from the mesenchymal connective tissue, and this mesenchymal connective tissue, it could be responsible for the formation of the cartilage, and later cartilage can be replaced by the bone or can be directly transformed into osteoblast, and this osteoblast is going to form the bone. If you remember the intramembranous ossification and the endochondral ossification. The teeth, the origin of the teeth is different, as embryonic origin is different from the bone. So that's why we consider the teeth is not part of the skeleton system. And usually the teeth consist of three materials. We have the material that found outside here. We call it the enamel. And then we have the bulk of the teeth, which is consist of the dentin. And also we have the area here where it's responsible for anchoring the teeth with the alveolar spaces that found in the upper and lower jaw, which we call it as the cementum. So we have three components: enamel, dentin, cementum. And if you remember from our talk, we said that the tooth, the short tooth or the brachiodent, consists of a crown, and then we have the area of the junction with the gum, we call the neck, and we have this part that embedded inside the alveolar space, we call it as the root. Usually at the area of the crown, we have the enamel and we have the dentin. At this area here, the root, we have just only the cementum and the dentin. The tooth, the component of the tooth, we have the bulk of the tooth is composed of the dentin. If you can see here, the dentin, we can see here at the area of the crown, the neck, and the root. However, only the enamel cover the area of the crown, and the area here, just below here, we have the cementocyte that only found at the area of the root. And we can see here there's the space here, we call it as the pulp chamber or the pulp cavity. Inside the pulp chamber or the pulp cavity, we have the pulp, which is the life material. If you remember also from my lecture before that I talked about the two types of tooth, we have the brachiodent and we have the hypsodent. The brachiodent as the teeth that we have in our body in a human, and most of the teeth in different animal species. These tooth or teeth, whenever they erupted from the gum, they cease to grow. They mean they'll never grow again. However, for the hypsodent, which is the long teeth, as the one that we can see in the horses, these one can grow, still can grow, and usually these tooth, when they grow, sometimes they cause and form sharp edges, and these sharp edges can hurt the animal because they can do hemorrhages inside his mouth, and sometimes they have difficulty in eating. This area here, the bulb, we have the area of the live tissue, and these usually contain the blood vessels and the nerve. And usually this area here, we call it as the root canal, because the blood vessels and the nerve will enter to the bulb from this area here. As I said before, that the most of the component of the tooth, which is the dentin, as I said, that the part that appear above the gum, we call it as the crown. At this area we have the neck, and the last part here we have the root. Inside here, as I said, we have the bulb cavity or the bulb chamber. As I said, the bulb is usually the live material, and usually these living material composed of the blood vessels, artery and vein, lymphatic, and also we have the nerve. Also we have the periodontal connective tissue that main function to anchor the tooth with the alveolar space, and these periodontal connective tissue serve as the periosteum because the cementum as a component, the main component of the cementum will be the waving bone. It's almost like the waving bone. You remember when we talk about the bone, that the first bone that form in our body, we call it the primary bone, and usually this bone is the waving bone and later can be replaced by lamellar bone. So this area here, we have the pulp, which is the area of the live tissue. We can see here, this is the area of the dentin, and we have here this area here where we have the periodontal ligament that anchor the tooth with the alveolar space. Uh, we talk about the component of the tooth. We saw it that we have the dentin. Mostly it's around 80% minerals and 20% collagen fibers. However, for the enamel, which is the covering layer of the tooth found at the crown area, it's composed of 98% to 99% of the minerals and 1% of collagen fibers. So the enamel layer is very strong hard tissue, but still brittle that can be easily broken. For the cells that are responsible for the formation of the teeth, we have three cells. We have the Odentoblast, which is responsible for the formation of the dentin. We have the cementocyte, which is responsible for the formation of the cementocyte, and we have the Immunoblast, which is responsible for the formation of the enamel layer. And you can see here the Immunoblast, they are temporary in the brachiodont because we said once the teeth is erupted, they will be lost. There will be no more formation of the enamel layer. So the first cell we have, which is the odentoblast, as we said that the bulk of the tooth is cellular. That's not composed of the cells. As I said, in the dentin we have 80%, in the enamel layer we have around 99%, which is minerals. However, for the collagen layers and the fiber cells, the percentage is limited. And the cells that are responsible for the formation of the dentin, which is the odentoblast, and usually these cells that you can see here found near the bulb, and they are going to form the predentin, which is only composed of the collagen fiber. Later there will be the addition of the and deposition of minerals to form the dentin. As you can see, whenever there is a formation of the predentin, there will be reduction in the bulb cavity. These cells, the odontoblasts, they are columnar cells. They are epithelioid because they don't have the basement membrane. These cells have the secretory appearance because, as I said before, they are responsible for the formation and secretion of the collagen. These cells have well-developed rough endoplasmic reticulum, and also they have a well-developed Golgi apparatus for the export of these proteins. As I said, the odontoblasts going to lay down the this area here we have the predentin, and we can see here that whenever there's a deposition of the predentin, there will be retreat and reduction of the side of the bulb toward the center, and these cells remain active throughout the life, so there's always a reduction in the size of the bulb cavity. The second cell we have, which is the immunoblast, again these cells are columnar, epithelioid, because they don't have a basement membrane. They are responsible for production of the collagen, the 1% that I said before, and usually after that there will be mineralization, as I said, in a percent around 99%. Very important about these cells, only remain or live only to the eruption of the tooth. Only when the tooth appear above the gum, these cells are going to be lost. So there will be no more formation of the enamel layer. As you can see here, this is the dentin, this is the amyloblast, and this is the pre-enamel. Once mineralized, we call it as the enamel. Again, these cells, as I said, remain until eruption. After that, they are going to be lost. There's two types of teeth. We have the milky teeth and we have the deciduous teeth, the permanent, the one that we are the first teeth that we have, we call it as the milky teeth or the temporary one, and usually after certain time in different animal species, they are going to be replaced with the permanent teeth, which is the deciduous teeth. And the deciduous teeth, there will be no more enamel formation. And as I said, there will be lost for the immunoblast, so there will be no more formation of the enamel. However, if we have remnant of the stellate reticulum, which is the layer just above the immunoblast before the eruption, maybe there will be growth of the tooth. But if we lose the immunoblast or reticulum, then we'll not have any growth for the enamel layer. As I said, some of the enamel could be trapped, and we can see these incisor teeth sometimes can grow, and that can hurt the animal, and they lost the ability to eat, and will be hard for them to be eating any type of food. Then we have the tongue. The tongue is very important function in eating, drinking, vocalization, and usually this organ composed of the muscle, and this organ is very mobile, can move a lot in different orientation. This tongue have dorsal surface and ventral surface, and usually the dorsal surface mostly keratinized stratified squamous epithelium, whereas the ventral portion will be thin keratinized stratified squamous epithelium. The tongue is sensitive and highly innervated, and there's a lot of blood supply in the tongue. In the tongue, we have special structures. We call it as the chemoreceptor sites, and these usually is the taste bud. The muscle in the tongue, we have the muscle in different orientation. We have three different orientation, and mostly each of these planes will be at the right angle to each other. That means if this orientation, then the other orientation will have the right angle, and then the other one going to have another orientation. So we have three different orientation, and between these different orientation, we have a right angle. See here, let's see, this is one orientation, and we can see here we have another orientation. All of these different orientation will allow the tongue to move and can adapt different type of movements. In the tongue, in the dorsal portion, we have the lingual papillae, and these lingual papillae mostly found in the dorsal surface of the tongue. See these structures here. All of these structures called the lingual papillae. As I said, they are special structures. These structures could be divided into mechanical papillae, and we have the gustatory papillae. Mechanical papillae responsible for direct the food toward the esophagus. After we eat the food or the animal eat the food, we need this food to be directed toward esophagus. And this mechanical papillae is responsible for this movement, the movement of the food, and direct the food to move from the oral cavity toward the esophagus. The lingual papillae, we have the other type, which is the gustatory papillae. And these gustatory papillae, they have a special structure, which is the taste bud, and these are very important because they can transform the into chemical stimuli into neural stimuli, and our brain can detect if we eat something that contains sugar or salt or sour. All of these will be the responsible of the taste bud. Mostly we have 6 different types of these lingual papillae, but the more common, which is the filiform, the fungiform, the valid, and sometimes we have the foliate. All of them are gustatory except the filiform. The filiform usually is the mechanical papillae. For the filiform, we can see is just a projection. This is keratinized stratified squamous epithelium, and they are covered the most of the time. They are keratinized projection. They are pointed caudally, so they can direct the food toward the esophagus. Their function as the ricks or the comb in grooming. As I said before, they have no sensory function. The second type we have the fungiform papillae, and usually they are large, and the shape of these almost like a mushroom, so that's why they call it as the fungiform, and usually they are elevated above the surface of the tongue. As you can see here, we have the epithelium, which is the thin keratinized stratified squamous epithelium. At the center here, at the core of these papillae, we have the connective tissue. As I said, they have the sensory structure, which is the taste bud. The second or the third type, we have the valid papillae. Usually they are very large, paired structure. They are found not as a single structure, they usually found as pairs, and usually they found at the area of the root of the tongue, and usually they don't elevate above the surface of the epithelium. Instead, they sunk below the surface, and usually they have numerous taste buds. And you can see here this space here that you can see between the epithelium where the fluid they are going to enter. This area we call it as moat, and we can see here just below this gland or structure papillae, we have the salivary glands. This is scanning electron microscopy. This is the valid, and we can see here this is the space. Usually, the material that we are going to taste, the chemical substance going to dissolve in saliva, and then this saliva going to enter at this area here, and then can be detected by these structures that found at this area here, which is the which are the taste bud. The fourth type we have the foliate, not found in all species. Sometimes we can see them in the lagomorpha, such as the rabbits. We can see them in sides of the tongue. We could not see it in the dorsal surface, usually found in the lateral surfaces. Because their appearance almost like a leaves of the book, they call it as the foliate. And very important, they contain this groove, and between this groove we have this structure here, which is the taste bud. For the taste bud, they are well-defined structure. They are the transducers of the chemical information to neural stimuli. They change chemical information into neural stimuli, and this is through the nerves going to the brain to decide the type of the material we already ingested. These taste buds usually sunk below the epithelium of the tongue. They're not found in the surface. They are found, you can see here, this is the epithelium, and they only sunk below the epithelium. And these taste buds contain the banana-shaped cells, and these cells, you can see here almost the appearance as the banana, and these cells contain microvilli because they have to absorb materials to be able to change it from chemical stimuli into neural stimuli. And you can see here at the distal portion we have the nerve axons. Between these cells which is the banana-shaped transducer cells, we have the supportive cells. And these supportive cells provide chemical and nutritional support, mechanical for support of these organs to keep them in their places, and also provide a nutritional support. These cells need a nutrient, and these cells can help provide these nutrition. As you can see here, this area here where we have this space here, we have the taste pores, and inside it we have the microvilli. Sometimes they call it as the taste hair. And any material to be able to be taste have to dissolve in saliva, then they have to be uptaken by microvilli, and these cells going to change them or transform them from chemical into neural stimuli. As I said, they are banana shaped. As I said, we have here, we have the microvilli, and at the distal end here, we have the axons of the nerves. As I said, we have the taste pores, and these microvilli, sometimes they call it as the taste hairs. As I said, these taste hairs are the microvilli. They have to absorb the chemical stimuli to be transformed into neural stimuli. Very important to know that the process of the taste is poorly understood. There's no clear information how we can taste things, how we can decide this is uh, salty or sour. All of this process is still unknown. So start to talk about the esophagus again. As apart from the digestive system consists of 4 layers: the mucosa, submucosa, the muscularis externa, and the serosa. Part of the esophagus will found at the neck area, and this area here we have the additional layer, which is the adventitia, which is the dense irregular connective tissue. For the esophagus, we have again the area, the epithelium, lamina propria. Then we have the muscularis mucosa. These are the 3 components of the mucosa. Then we have the area of the submucosa. Then we have the area of the muscularis externa. As I said, we have the area of the serosa. The esophagus is the muscular, flexible, distensible organ. The wall of the esophagus, we have 2 types of muscle. In the muscularis mucosa, we have the smooth muscle, whereas in the muscularis externa, depending on the animal species. For example, in the dogs and ruminant, we have from the entire length of the esophagus, from the beginning toward the area that we reach the stomach, the wall of the esophagus will be from skeletal muscle. So these animals can easily vomit and do rumination. Vomiting for dogs, rumination for the ruminant. Whereas in human, horse, cats, about 4/5 of the total length of the esophagus is consist of skeletal muscle or composed of skeletal muscle, whereas the distal fifth is composed of smooth muscle. So these animal, human and this animal, horse, cat, they cannot vomit. For the mucosa, the epithelium will be thin keratinized to keratinized stratified squamous epithelium, depend on the diet of the animal. Then we have the area of the lamina propria. Then we have layers from the smooth muscle in the final component of the mucosa, which is the muscularis mucosa. Then we have the area of the submucosa, where it have the gland the esophageal gland or the submucosal gland. And if you remember in the early slides, I said that the glands that found in the submucosa only found in the area of the esophagus and the duodenum. Then we have two layers from the muscle, the inner circular and the outer longitudinal. As I said, they are composed of skeletal muscle from the beginning to the end of the esophagus for the whole length of the esophagus, whereas in cat, human, and horses, the distal 1/5 will be composed from smooth muscles. So we can see here, this is the area of the epithelium, thin, keratinostratified, then we have the area of the lamina propria, and then we have the area of the smooth muscle. These three components are the mucosal layer, and then this area here just below the muscular mucosa, the area of the submucosa. As I said, there is a gland in the area of the submucosa. Again, this is the epithelium, muscle, and we can see here this is the area of the submucosa where we have the esophageal gland or the submucosal gland, and usually these and the secretion of this gland will be mucus, and that will help in lubricating the food and facilitate the movement of the food from the esophagus toward the stomach. Then here we have the area of the muscle. As I said, we have two layers, the inner circular and the outer longitudinal layer, which consists of skeletal muscles. So this is the area where we have the muscularis, mucosa, then we have the glands. All what we can see here are the glands, and this area here where we have the skeletal muscle or the muscularis externus. See here we have the muscles, and most of these muscles, skeletal muscle, we can see that the nucleus is found in the periphery, which is the characteristic feature of the skeletal muscle, that the nucleus is found in the periphery. Again, all of these muscles are the skeletal muscles, and you can see here the nucleus is found in the periphery. Now we'll talk about the esophageal gastric junction, which is the area where the epithelium of the esophagus is changed into the epithelium of the stomach. Usually the epithelium that found in the esophagus, keratinostratified squamous epithelium. When it changed to simple columnar, that means now that we are in the area of the stomach. Usually this change will be abrupt transition. That will be abrupt transition that we can directly see that change from stratified squamous epithelium into simple columnar epithelium. And usually the location is varies with the species. For example, in dogs and cats, usually this area of the esophageal gastric junction is found at the distal end of the esophagus before the entrance to the stomach. Whereas in the horse and pigs happens at the area, this junction will be inside the stomach, and usually there will be like a line separate the non-glandular by this non-glandular, which is the stratified squamous epithelium, from the glandular portion with the simple columnar epithelium, and there will be like a line separate between the glandular and non-glandular portion. So as we can see here, in the dogs and cats will be in the area of the esophagus, whereas in the pigs and horse will be inside the stomach. In the ruminant, we know that the stomach is composed of 4 compartments. We have the rumen, reticulum, mesum, apomesum, and we know that the first 3 components, the epithelium, will be Keratinized stratified squamous epithelium, whereas the abomasum is the only part that have the glandular portion. So again, the change in the epithelium will be inside the stomach. As you can see here, this is the epithelium, which is the epithelium of the esophagus, whereas here will be the epithelium of the stomach. Now I'm going to talk about the stomach. The stomach, as we know, that's the appearance is the J shape. Usually this is the area of the entrance of the esophagus. We call it as the cardia, and this is the exit to the duodenum. We call it the pylorus. And we have the sphincter here that control the food that exit from the stomach to the duodenum. We call it as the pyloric sphincter, whereas the food that enter through the esophagus toward the stomach will be through the cardiac sphincter. As we can see here, they divided into cardia. Then this is the area of the blind sac. We call it as the fundic. Then we have the body, and finally we have the pyloric region. All of these areas are the area of the stomach: cardia, fundus, body, pyloric. For the histological appearance of the stomach, again they are consist of four layers. We have the mucosa, submucosa, muscularis externa, and serosa. The epithelium that found in the stomach will be the simple columnar mucus producing cells. Again we have the area of the lamina propria, and then we have the area of the muscularis mucosa, then we have the area of the submucosa, and then we have the area of the muscularis externa. And here the muscularis externa is consist of three layers: inner oblique, middle circular, and outer longitudinal. And the outermost layer will be the serosa. As I said, we have the cardiac region, we have the pyloric region, and finally we have the fundic region. For the histological appearance, the cardiac region and the pyloric region, they are almost similar, and sometimes is difficult to differentiate between the cardiac and pyloric. However, there is a difference in the fundic region, because in the fundic region we have special cells that only found in the fundic region. That's the parietal and the chief cells, and we are going to talk about them later. As I said, we have the mucosa, submucosa, muscularis externa, and we have the serosa. This is the area of the stomach, and we can see here this is the epithelium, which is the simple columnar mucus producing cell, and then we have the lamina propria, and then we have the muscularis externa. Inside the stomach we have the glands. These glands we call it as the gastric glands, and these glands open into the surface of the stomach through special opening. We call them as the gastric pits. Then we have the area of the submucosa. Then we have the muscularis externa. As I said, it consists of three layers: the inner oblique, middle circular, and the outer longitudinal. And finally we have the serosa, again, which is composed of the simple squamous epithelium. If we start with the mucosa, again will be the epithelium that we can see here. This is the simple columnar. We call it as mucus producing cells, because these cells are responsible for production of a lot of the mucus, and this mucus is very important because it's going to protect the stomach from the acid and enzyme that produce inside the stomach that can burn and digest the mucosa. Just beneath the epithelium, which is the simple columnar mucus producing cell, we have a layer of the loose connective tissue. We call it as the lamina propria, and again this layer contains the typical cells of the connective tissue. we have lymphocyte, plasma cell, mast cells, and we have the fibroblast. And finally, we have the final component, which is the muscularis mucosa. Again, it will be poorly defined smooth muscles, and usually they divide it into two layers, inner circular and outer longitudinal. But they will not be as clear as we can see it in the area of the muscularis externum. As I said, the epithelium that we have here, we call it as the simple columnar mucus producing cells. These cells will contain a lot of the mucus granules that found in an active form. We call it as mucinogens. Once secreted, they will be form the mucin. And usually the secretion from these cells will be cloudy, thick, viscous mucus, and it will contain high amount of or content of bicarbonate. And this is very important because this bicarbonate will protect against the acid, as we know that inside the stomach there is production of the hydrochloric acid, and this hydrochloric acid can burn the stomach. Very important thing about this lining epithelium, it's renewed every 3 to 5 days. All the epithelium that found in the stomach going to replaced totally by a new epithelium, because these cells usually exposed to the enzyme pepsinogen or pepsin and hydrochloric acid, which can damage these epithelium. So this is the epithelium, simple, columnar, and you can see the cloudy appearance in the apical portion, and these usually are the mucinogen. Just beneath the mucosa, we have the area of the submucosa, and usually this layer is a dense, irregular connective tissue, again contain the typical component of the connective tissue, which is the fibroblast, mast cells, lymphocyte, all of these cells. Also, they will contain nerve plexuses that are responsible for the movement and control the movement and the contraction of the smooth muscle that found in the area of the mucosa, which is the muscularis mucosa. We call it as the Meissner's plexus. For the muscularis externa, it's composed of 3 layers. As I said, we have inner oblique. Usually this one is incomplete. That means it's not found in all the whole structure of the stomach. Usually they are well developed in the areas, for example, pyloric or in the cardiac region. The second layer we have, which is the middle circular, and finally we have the outer longitudinal. Very important, the middle circular layer at the area of the cardiac region the cardia, they are going to be thickened, and similarly in the area of the pyloric region. And these two structures are very important because they were responsible for the formation of the cardiac sphincter, also the pyloric sphincter, which control the entrance of the food toward the stomach, also the exit of the food from the stomach toward the duodenum. Finally, we have the final layer that found at this area here, which is the serosa, which cover the stomach, and usually it's simple squamous epithelium. The other important structure that found in the stomach, which is the gastric gland, and usually these gastric gland is found at the area of the layer, which is the mucosal layer. And this gland is the basic secretory unit of the stomach. They're responsible for the secretion that produced in the stomach. As I said before, there's a production of the hydrochloric acid, also for the pepsinogen, which is responsible for the chemical digestion of the food. Usually these glands found in the three areas of the stomach, in the fundic, pyloric, and the cardiac, and usually the glands that found in the cardiac and pyloric region, they are similar. However, they are different from the gland that found in the fundic region. Usually these glands, they divided into neck region, then we have the body, and the distal portion, we call it as the fundus. And usually the opening here, the opening that's found here, we call it as the gastric pits. Usually these gastric glands will contain different cell types. They will contain parietal cells, the chief cells, the mucous neck cells, the enteroendocrine cell, the regenerative cells that are responsible for the regeneration of the whole epithelium that found in the stomach epithelium. As I said, these glands, you can see here, this area here is the area, neck, body, and the fundus, and the opening here, we'll call it as the gastric pits. Usually we have different cell types that found at the gastric gland. We have the gastric neck cells, we have parietal, we have chief cells, enteroendocrine cells, and we have the regenerative cells. And each of one of these cells have a certain function. So we can see here, this is the surface epithelium, which is the mucus producing cells, and you can see here the mucinogen, and then we have the gland, we have the regenerative cells, we have the mucous neck cells, we have the oxyntic or parietal cells, we have the zymogenic cells or the chief cells, and finally the cell that found in the bottom of the gastric gland, which is the enteroendocrine cells. This is what we are going to see under the microscope. We're going to see this epithelium, which is the simple columnar mucus producing cells, and this is the area here where we have the muscularis and mucosa. From here to here, all of that area, which is the area of the gastric gland, and the opening that found at this area here, this will be the opening of the gastric pits. And the cells that we can see here, all of these cells are the cells of the gastric gland, and between these cells we have the area of the connective tissue, which is the area of the lamina propria. Here, this is the epithelium, and this is the opening of the gland, which we call it the gastric pits. This is scanning electron microscopy. Again, the lining epithelium, which is the simple columnar mucus producing cells, and we can see here, this is the opening of the gland, which is the gastric pits. As I said, we have glands in the cardiac region, pyloric region, and the fundic region. As I said, this gland will extend from the surface epithelium toward the layer of the muscularis mucosa. As I said, they're responsible for production of the mucus, and this mucus is very important in protecting the stomach against the hydrochloric acid and pepsinogen that are produced by the cells that found in the fundic region. So in the fundic region, we can see here, again, we have the surface epithelium, we have the gastric gland, but very important, in the fundic region, we have the oxyntic and the zymogenic cells, the parietal cells, and the chief cells. The parietal cells is responsible for production of the hydrochloric acid, whereas the zymogenic cells are responsible for production of the pepsinogen. These cells, the parietal cells and the chief cells, are only found, very important, they are only found in the fundic region. So again, this is only the area of the mucosa. You can see the epithelium that found at this area here, which is the simple columnar mucus producing, and we can see here From this area to this area, we have the gastric glands, and between these gastric glands, we have the area of the lamina propria. See here, the whole length, we have the glands. We have two types of cells that only found in the fundic region, which is the chief and parietal cells. These chief and parietal cells will not find them at the area of the cardiac region or the pyloric region. So it's easily we can differentiate between the and cardiac region from the area of the fundic, because in the fundic we have the chief and we have the parietal cells. However, in the area of the cardiac and pyloric region, there is no chief and parietal cells. So we can see here, this is high magnification image for the gastric glands. We can see here we have two different types of cells. This cell here, where the cytoplasm is clear and eosinophilic, these cells or these cells are the parietal cells. However, these cells that found and they are darker and almost they are cloudy in appearance, these cells are the chief cells. The chief cells, or the other name, which is the zymogenic cells, they are pyramidal in shape. These cells, as I said, responsible for protection of the pepsinogen. Once released from the cells, going to be activated and change or activated to pepsin. Also, there is a production of renin, and also there is production of lipase. And these cells, they have abundant rough endoplasmic reticulum, they have supranuclear Golgi apparatus, because there's a production of these proteins, and these proteins need well-developed small rough endoplasmic reticulum, also need well-developed Golgi apparatus. You can see all of these cells here, all of these cells, the darker cells, these cells are the chief cells. On the other hand, we have the parietal cells. These cells, very important because they are responsible for production of the hydrochloric acid. These cells, I can see here, all of these cells, parietal, mean that they found in the wall of the glands. These cells, again, they are pyramidal in shape. They have vast number of the mitochondria. These cells consume a lot of oxygen. See the difference. These cells are the parietal cells, whereas these cells are the chief cells. As I said, the parietal and chief cells only found in the fundic region. As I said, the parietal cells, the pyramidal in shape, as I said, they are responsible for production of the hydrochloric acid and also produce intrinsic factors that are needed for vitamin B12 absorption in the ileum. So again, this is the parietal cells, whereas the cells that we can see here, all of these cells are the chief cells. This is the pyloric glands. As you can see in the pyloric, almost like the cardiac region. However, the pyloric glands don't contain the chief and parietal cells. The other cells that found in the area of the gastric gland, which is the enteroendocrine cells, usually these cells is scattered in the area of the fundic region of the gland, which is the distal portion of the gland, and usually these cells polarize to the bottom. That means that the secretion of these cells will be released from the cells and go to the area of the lamina propria, which is going to be absorbed by the blood. These enteroendocrine cells, almost there's like more than 20 types of these endocrine cells, and each of one of these enteroendocrine cells responsible for production of the hormone that responsible for activation or inhibition of the digestion process. For the muscularis externa, we have three layers. We have the we have the inner oblique, middle circular, and we have the outer longitudinal. So we can see here, this is the middle circular, and out here we have the outer longitudinal. And the layer that we can see outside here, this is the area of the serosa, which is the simple squamous